Confocal Microscopy by Sidra Aziz. Some background information on confocal microscopy. Confocal microscopy was a concept that was developed in 1957 by Marvin Minsky. A new technique was necessary because the conventional microscopy was a in-focus and out-of-focus light, which resulted in blurry images of the out-of-focus light. The new technique asked for in-depth of field view, which are short and usually measured in microns. The main components of confocal microscopy include light source, dichroic mirror, objective lens, specimen, confocal pinhole, and a detector. The only difference in confocal microscopy from conventional microscopy is the confocal pinhole. Confocal pinhole blocks out-of-focus light from being detected, so a clear in-depth image is visible. Light source hits a wavelength off of dichroic mirror, which is nothing but a piece of glass with metal that bounces wavelength off through an objective lens that hits the specimen. Another wavelength bounces off of this specimen through dichroic mirror into confocal pinhole and hit the detector, where we can see our image. A paper that used confocal microscopy is titled Assessment of Human Pancreatic Islet Architecture and Composition by Laser Scanning Confocal Microscopy. In this paper, the research that was conducted was to better understand the quality and characteristics of human islets used for transplantation. They performed detailed analysis of islet architecture and composition using confocal laser scanning microscopy. Human islets from 6 separate isolations provided by 3 different islet isolation centers were compared with isolated mouse and non-human primate islets. The authors wondered if an isolated murine islet would have alpha and delta cells reside at the periphery of the beta cell core. The experimental design of the figure is the representative series of optical sections through an isolated mouse islet. The figure shows that a series of optical sections through the entire isolated mouse islet was acquired at 1 m intervals in the axial z dimension. Individual optical sections demonstrate absence of non-cells in the islet interior. The stack of optical sections was 3D reconstructed. In this article, the researchers were able to conclusively demonstrate that normal adult marine islets have a beta cell core and non-beta cell mantle. My question is, how long does it take for all the images how long does it take to take all the images and make a 3D structure from the stock of images?